Okay, now we're going to talk about immunopathologies, and we're going to start with hypersensitivities. So there are 4 types of hypersensitivities that I want you to know. So first one will be very creatively named type 1 hypersensitivity. It is an allergy. It's also called immediate hypersensitivity. So what is the actual mechanism for an allergy? So I'm going to use the same acronym AG that I used for antigen before. I'm going to use it here for allergen. Is that okay? Okay. So you get exposed to allergen, and your immune system, your B cells, produce immunoglobulin E. Okay? Now, immunoglobulin E gets in the bloodstream and eventually binds to mast cells and to less extent to basophils. Okay? That makes sense? So IgE is bound to mast cells. This is your very first exposure. Look at this, nothing happens. You see, nothing happens here. Maybe it's going to be your first and second and third exposure, but at some point, some subsequent exposure, you get exposed to allergen again. And now allergen are going to bind to immunoglobulin E, which is, as you remember, bound to mast cells. And now this binding of allergen to immunoglobulin E will result in mast cell degranulation. Does that make sense? So mast cells will release cytoplasmic granules that contain bradykinin, histamine, and you will get your typical reaction. Depending on where those mast cells are, you can have runny nose, you can have itchy eyes, you can have swelling of the smooth muscles, and asphyxia, you can have hives, all those reactions. Make sense? Now, this scheme implies that at some point you can exhaust B cells that produce immunoglobulin E. So basically what I'm trying to say is that so I'm allergic to cats. We can get the cat at home, and I'm going to immensely suffer for I don't know how long. But eventually this will end and my allergy will go away, or the cat will go away, but that's a different story. I'm not ready for this commitment, but you see where I'm going. And it was shown that for many allergy cases, the exposure to very, 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 very small amounts of antigen can actually improve the tolerance. There was a study showing that children with peanut allergy, when they get exposed to the protein in peanut butter that they're allergic to, when they receive extremely small amounts under the medical supervision, it increases their tolerance, not to the degree that they can eat PBJ, but to the degree that they can be at the same table with someone who eats PBJ. Does that make sense? So not like at imminent risk of death every second of the elementary school. Okay? The allergy part. Clear? Okay, now let's talk about a really exciting story, which is type 2 hypersensitivity. And it is mediated not by immunoglobulin E, but immunoglobulin G. And this is the story about transfusion reactions. Before Karl-Heinz Steiner discovered 4 main blood types, Landsteiner blood group. The joke was that for blood transfusion, you need 3 sheeps. You need a doctor, a patient, and an actual sheep. Okay, because people transfuse the blood from all kinds of different crazy sources. So Landsteiner blood groups are based on different agglutinogens and agglutinins. Should be 2 G's here. So agglutinogens are antigens on the surface of red blood cells. So they can be absent. This is O blood group. It can be agglutinogen A, B, or A and B both. By the way, that's a great example of incomplete dominance in non-Mendelian inheritance. So people have alleles, may have no alleles, no A, no B, may have only A allele, may have only B allele, or may have both A and B alleles, and in this case they can be AB blood type. Does that make sense? So those are antigens. They are very, very complex carbohydrates. Now, interestingly enough, in the plasma, people have antibodies, and people with a blood type O have antibodies both against A and against B. Here, only against B. Here, only against A. People with the blood group AB have no agglutinins. Does that make sense? So basically, agglutinins and agglutinogens are mutually exclusive. Now, how does it translate to the blood transfusion? You probably know it already, just a reminder. So when you transfuse blood, you need to look at the red blood cells of the donor and the plasma of the recipient. Because during the transfusion, plasma of a donor, just such a small fraction that it's not really that important. Does that make sense? So for instance, you try to transfuse AB red blood cells to someone. Who can receive AB red blood cells? Only AB. Because if you mismatch blood types and you transfuse it to someone else, the agglutinins will bind to agglutinogens, which will result in the agglutination of red blood cells, hemolysis, and kidney failure, and vascular obstruction, and so on and so forth. Does that make sense? So at the end of the day, the universal donor is type O. The universal recipient, because they have no agglutinins, are type AB. Does that make sense? Now, there are something like 200 different blood groups. So, a whole bunch of different surface antigens on red blood cells. So we're not going to talk about all of them. But we're going to talk about another really important antigen, which is rhesus factor, or D antigen. So what I want you to fully appreciate is that D antigen and Landsteiner blood groups, blood types, they are independent. Does that make sense? So these are two different classifications. Sorry. So people who have D antigen on their red blood cells called rhesus positive, and people who don't called rhesus negative. Okay, so for instance, let me explain what I mean by the independent. I am personally O positive. Okay, so it means I have no Landsteiner agglutinogens, but I do have the antigen on my red blood cells. Someone who is AB positive, they have A agglutinogen, B agglutinogen, and D antigen on their red blood cells. Is that clear? What's important is that there are no pre-existing antibodies against D antigen in rhesus negative folks. Only if a person who's rhesus negative receives rhesus positive blood, they will eventually develop the antibodies. Does that make sense to you or not? So the immune system will recognize the unknown, the antigen, as a bad one, and will produce immune response to it. We good? That makes sense? Okay, simple example. Let's presume I am O positive. You are O negative. I have no idea what your... Okay, O negative. Excellent. So you are O negative. So if you will give me your red blood cells, for my immune system, there is nothing in them. It's fine. My immune system doesn't see them. Does that make sense? If I give you mine, your red blood cells look like this. My red blood cells look like this. Your immune system sees that D antigen, does not recognize it as self, and starts making antibodies to it. Does that make sense now? So first time, you should be fine. But if you try to receive another bout of my blood later, you're going to have antibodies that will attack my red blood cells, will cause hemolysis, and you're going to be in trouble.
So is that, like, so when I had my children, I had to get the RH shots?
That's exactly what it is.
Yeah. So here's the thing. Technically speaking, I am being O positive. I'm not a universal donor. I can donate my O positive blood to anyone with any Landsteiner blood group who also is rhesus positive. You with O negative blood type is truly universal donor. Your blood can go into anyone. Does that make sense? Now, this rhesus factor and the fact that people can mount an immune response to it explains the cases of rhesus conflict between mother and a child. So imagine that you have a mother, well, strictly speaking, mother and fetus. You have mother and fetus, and they are separated by placenta. Okay? Makes sense? Their bloods don't mix. That's important. Mother is rhesus negative. Child is rhesus positive. It's fine. That's the first pregnancy. It's fine. Bloods do not mix. Clear? Now, during the delivery, placenta detaches, and for a very short period of time, fetal blood gets into the maternal circulation. Okay? So just as we discussed a minute ago, maternal immune system locates this Rh plus red blood cells and mounts an immune response to them. Okay? But it's fine. It's a first pregnancy. Child is gone. I mean, child is born. Like, child's not there, so it's okay. Good? And now you've got second pregnancy. And you've got the same situation. Mother being rhesus negative and child being rhesus positive. Well, fetus, not a child yet. Okay? But the problem arises from the fact that those anti-Rh antibodies that are circulating in mother's blood are immunoglobulins G, and they can cross placenta. And they will attack red blood cells of a fetus. And they will result in what's called erythroblastosis fetalis, or hemolytic disease of newborns. Does that make sense? It's not a great story. What can you do? And the mechanism is absolutely stellar. I just love that story. So imagine we go back to square one. We go back to a mother, first pregnancy, right? So what this mother will receive, a so-called ROGAM shot, which in brief is anti-rhesus antibodies, but they're recombinant antibodies. Those antibodies can't cross placenta. Does that make sense? No?
Isn't it the
Anti-Rh,
antibodies that you just said did cross the...
naturally formed, can.
Okay.
Those are artificial antibodies, and they are modified, so they can't.
Did they then block the natural ones from occurring?
Now look how they did that. So a woman has those artificial antibodies in her bloodstream, okay? Delivery, placenta detaches. And those artificial antibodies attack immediately, immediately attack red blood cells of a fetus. They attack them so fast and destroy them basically so fast that immune system of a mother doesn't see them, doesn't have time. Does that make sense? It's absolutely just super elegant mechanism. We're good? And honestly, if mother is rhesus negative, nobody bothers to test the child. Because nobody's going to poke fetus with a needle just to figure out what the rhesus factor there, right? Just program shot and that's it. If fetus is rhesus negative, big deal. Okay? Make sense? Now, third type is also mediated by immunoglobulin G, and this is the truly autoimmune diseases mechanism. It's a formation of immune complexes. Okay, so remember when we were talking about B cells, we mentioned that they undergo negative selection to remove ones that are self-reactive. So imagine they were not removed. Now you have self-reactive B cells. Anti-self antibodies are produced. So these anti-self antibodies, in case of usually those immune complexes, are formed in basement membranes. So I'm going to pause here for a second. So these antibodies, check this out, these antibodies will target proteins that comprise basement membranes in the epithelium. Does that make sense? We're good? So now imagine this, you have those immune complexes, a whole bunch of antibodies sticking to self-antigen. It's not supposed to be, it's wrong, okay? They're sitting in the basement membranes, and your phagocytic cells, neutrophils and macrophages, discover that something is surrounded by antibodies. Remember, antibodies tag things that must be destroyed, right? So neutrophils and macrophages start to infiltrate your epithelium, trying to destroy the immune complexes. What it results in? Inflammation in the epithelium. If it's a skin, it's a psoriasis. If it's a blood vessel, vasculitis, which may result in thrombotic events. If it's glomerular capillaries, glomerulonephritis. If it is synovial membrane in the joints, arthritis synoviitis. What I just listed, thrombotic events, arthritis, vasculitis, skin lesions, glomerulonephritis are typical symptoms of the condition known as systemic lupus erythematosus. Classic type 3 immune hypersensitivity. Does that make sense? Okay, good, awesome. And finally, the last one, creatively named type 4 hypersensitivity. It's a delayed hypersensitivity mediated by T cells. So in this case, you start with an antigen exposure, okay? And this particular antigen will stimulate memory T cell response. Now, what I want to highlight at this point, when you are exposed to that antigen first, nothing happens. You know, you don't have any particular reaction. You just form a pool of memory T cells. Okay? Then you get exposed to that same antigen for the second time. Okay? And you have T cell activation. If you remember something from our last conversation about T cells, you're going to have both CD4, the helpers, and CD8, the killers, activated. Okay? So killers will do what they can. They're going to kill cells that bear this antigen. And CD4 cells will stimulate specifically macrophages and also target them on those same cells. So you're going to have macrophage and cytotoxic lymphocyte toxicity. And that will result in whatever condition you're experiencing. If the pure example of type 4 hypersensitivity is TB test, so exposure to tuberculosis leads to formation of the pool of memory T cells. When you get an intradermal injection, if you have those T cells, you will produce a little bump, which is essentially macrophage and cytotoxic lymphocyte cytotoxicity. Does that make sense? Another classic example is poison ivy. First exposure does not result in anything, it's the second one that gives you all those terrible blisters. Make sense? Good? This is what?
The smallpox vaccine.
Smallpox vaccine is just the effects
How
of...
it reacts to, because I received it.
In the army?
No,
No.
the Coast Guard.
Huh?
The Coast Guard.
So, military.
Yeah, military. I don't know, it was gross.
Smallpox vaccine, so it's not type 4 hypersensitivity, it's just the effect of the weakened virus. Smallpox vaccine is a true example of the, I think, proper approach to public health. So I had the privilege to attend a talk from a person who was responsible for elimination of smallpox in Soviet Union. And they basically said that when they had first batch of vaccine, it was very reactogenic, and they had 1 per 100,000 cases of encephalomyelitis, basically inflammation of the brain, that led to paralysis or death. And they thought, no, 1 per 100,000 is not good. And they kept on improving the vaccine and ended up with 1 per 200,000. And they couldn't move any further than that. And at some point they said, that's good enough. This is the price in human lives that we can pay for eradicating the disease. And they knew that 1 out of 200,000 people vaccinated will develop the pretty horrible disease. Nevertheless. So yeah, it's just the virus, you know. So again, kind of match the description of the mechanism and the type. That's basically it. Okay.